Program Bachelor of Science, 3rd year, subject Botany, semester 6, course code BOC 109, course title Molecular Biology and Genetic Engineering. Title of unit Gene Structure, Regulation and Modification of RNA. Module name Eukaryotic mRNA Processing and Stability. Outline Eukaryotic mRNA Processing and Stability. Learning outcomes describes pre-mRNA, elaborates steps of mRNA processing, explains 5' capping, explains mechanism of addition of 3' poly A tail. Eukaryotic mRNA processing and stability. Many of the RNA molecules in bacteria and virtually all RNA molecules in eukaryotes are processed to some degree after synthesis. A newly synthesized RNA molecule is called a primary transcript. These primary transcripts range from 500 to 50,000 nucleotides. Pre-mRNA or hnRNA. Pre-mRNA remains confined to the nucleus and is called heterogeneous nuclear RNA or hnRNA. The hnRNA molecules which are destined to produce functional mRNA undergo RNA processing. Eukaryotic pre-mRNA receives a 5' cap and a 3' poly A tail before introns are removed and the mRNA is considered ready for translation. mRNA processing. So during mRNA processing, it basically involves the primary RNA transcript which is being processed and it produces the mature RNA. The primary RNA transcript, which includes the coding, that is the exons, and the non-coding regions, which includes the introns. So during RNA processing, the non-coding regions, also called as the introns, they are spliced out and it produces the mRNA which will include only the exons. During RNA processing, there also takes place the addition of 5' cap. So this 5' cap will be added at the 5' end and the poly A tail will be added at the 3' end. First, the 5' capping. A 7-methylguanosine cap is added to the 5' end of the pre-mRNA by a phosphate linkage while elongation is still in progress. The 7-methylguanosine cap contains an unusual 5' to 5' triphosphate linkage and 2 or more methyl groups. The 5' cap protects the nascent mRNA from degradation and assists in ribosome binding during translation. Structure of 5' cap. So this picture depicts the structure of 5' cap, that is the 7-methylguanosine which is being attached to the 5' end of mRNA via the triphosphate bridge, also called as the phosphodiester bond, which is being formed. 5' capping. During 5' capping process, the pre-spliced or the immature mRNA called as the pre-mRNA, it will undergo RNA processing, which includes the addition of 5' cap, that is the 7-methylguanosine which will be added at the 5' end. After this addition, there will be a removal of introns by the splicing process. And we have the mature mRNA which is being produced, which will include 5' cap. Second, addition of poly A tail. A poly A tail is added to the 3' end of the pre-mRNA once elongation is complete. The poly A tail protects the mRNA from degradation and aids in the export of the mature mRNA to the cytoplasm. It is also involved in binding proteins involved in initiating translation. Poly A tails are added to the 3' ends of transcript by the enzyme called as poly A polymerase. The 3' end substrates of poly A polymerase are produced by endonucleotic cleavage of the transcript downstream from the polyadenylation signal AAU-AAA. After cleavage, poly A polymerase adds tracts of adenosine monophosphate residues about 200 nucleotides long to the 3' ends of the transcript. Polyadenylation. So this picture basically depicts the addition of poly A tail at the 3' end. During the first stage, that is during the elongation, there will be a synthesis of a pre-mRNA. So this pre-mRNA, there will be a endoclivatic cleavage which will occur due to the endonuclease enzyme at the conserved sequences called as AAU, AAA. So this endonuclease will go and bind to these conserved sequences and it will cause a cleavage. Next is the polyadenylation process, wherein the enzyme polyadenylate polymerase, it catalyzes the reaction and it adds the nucleotides adenosine to the 3' end on the pre-mRNA. So this is the polyadenylation process which occurs. References. Thank you.